I'll give it a few minutes. Thank you all for joining. Just wait maybe 1 or 2 minutes just to make sure everyone is admitted. Welcome to the Expansion Microscopy user group. So we might just get started. First of all, for people that don't know me, my name is Gabriela Siegel. I'm part of the Biological Optical Microscopy platform at Melbourne University. And I've been organizing these Australian hosted meetings for the Expansion Microscopy group. And the idea of these meetings is to sort of get people together to discuss about this topic and help each other. We are organizing these meetings together with the Canadian counterpart. So all these Google documents you'll see in the chat here have also been filled in, and you can see some of the information the people in Canada have been filling in. If you have any questions, then people should be able to put on a question and make sure you put an email contact so people can contact you if they have a specific answer for you. And all of those documents are really embedded in the last document, which is the master document. So we'll take you to all 3 of them. For today also, as I was saying before, we would like to have attendance. So make sure you add your name into this attendance list. I think that's all pretty much I wanted to say for now. For today, we have scheduled a presentation from Alina. She's joining us from Israel at the moment, so she woke up really early to share some of her results, so that's really nice for us. And for future meetings, if people want to volunteer, you can write it on the chat. We also can do it via the Google document, but people can just contact me as well. I guess we'll let Elena talk and then we can have discussion, and feel free to put things on the chat and questions you have. So I'll let you.
Okay. Hi, everyone. So my name is Alina. I'm from the Weizmann Institute, Israel. And at our last meeting that was held a couple of months ago, I guess I was the only one who had much experience in expansion, so I wanted to share it with you and just to show you some techniques that I've been using. So this lecture will be a bit technical, so just with some tips, what you can do to improve this technique or to use it to apply to your samples. So it will be like a quick overview of several techniques, and feel free to contact me or ask questions during the presentation or by the end. So I'll just share my presentation. A second. Sorry, didn't start from the beginning. Okay. Okay, so I work on the Drosophila testis, and in this image you can see one of the expanded samples. You can see dividing cells. So the blue is the nucleus, and the purple one, like the pinkish one, is the mitochondria. I want to.
Sorry, I don't.
Sorry,
seeing your screen.
You don't see my screen. Oh, sorry.
We were seeing it before.
Oh, This one. Okay. Now you see it. Do you see it now? No.
Oh, maybe it's just me. Sorry.
No. I'll try again.
People are saying they can see it, so maybe I just did something wrong.
Gabriela, do you see it now?
Yeah, I think that's better. Yeah. Okay.
Okay.
Yeah.
Okay. So as I was saying, here you can see two dividing cells. The blue is the nucleus. It's just a 3D reconstruction done by Marek's software. So it's the blue is the nucleus, and the pink one is mitochondria. So I would like first to introduce my system. I work on Drosophila testis. This is the schematic representation of Drosophila testis. So this system is really nice because the differentiation of the sperm occurs as it progresses along the testis. So here's the niche of the stem cells. Each unit of the stems of the germ cells is surrounded by cyst cells, somatic cyst cells. They divide synchronously for mitotic divisions. Here you can see that the early stages of the spermatogenesis. So they start as a primary spermatogonium. They start to divide, form meiotic divisions, finishing by these 8 interconnected large cells called spermatocyte. And this stage, the cells aggregate, the cells accumulate a lot of material, and they grow in size and enter meiosis. So they go through 2 meiotic divisions. And after that, we end up with 64. After 2 meiotic divisions, we end up with 64 interconnected round spermatids. And in this stage, something really unique happens. All the mitochondria aggregate next to the nucleus, and they start to fuse, forming this giant sphere mitochondria. It's almost 6 microns in diameter. And this sphere is called the Nebenkern. Nebenkern in German is next to the nucleus. And then this Nebenkern is composed of these 2 mitochondrial masses wrapped around each other. And it has like these concentric circles of membranes. So this stage also termed an onion stage because it resembles an onion in cross section. And then these 2 masses start to elongate along the sperm tail and separate from each other. And by the end, we have this mature sperm 2 millimeters long with 2 millimeter long mitochondria. So as you can see, these images, they were these drawings were based on the actual EM images, and this process was described in ultrastructural details even through 70s by Tokayasu and his team. So, but the main question of my interest was the mitochondrial fusion, this process, how this process happens. So in order to get better insight in this process, I applied several microscopy techniques. I used EM, but EM is really hard to perform, and you need to fall exactly in the correct slice, and you get only like a thin section that doesn't represent the whole tissue. So I turned to the expansion microscopy. Expansion microscopy is really a nice technique that allows you to use a regular confocal or any other fluorescent microscope to visualize your samples. And it gives you much better resolution. So how it works, actually the sample is just physically expanded. In this cartoon, you can see like a represented, let's say this is the sample, so it's expanded 4 times and you can see much more details. And there are several expansion techniques available now. You can find any different kinds of techniques online, and every year we have new techniques rising. So I'll give you a short overview of 3 techniques that I have experience with. So let's start first with the main principle of the expansion microscopy. So first you have your sample, you fix it, you embed it into gel, you digest it using either chemical digestion and denaturation by heat or enzymatic digestion, and then you add water and your gel swells and the sample expands. And then you can visualize the sample using a confocal or other fluorescent microscope. So the first protocol dealing with expansion microscopy was this one. Sorry, it was this one, but then this is like the more modern one, the newest one. So in this technique, actually, you stain your sample before you start the expansion protocol. So the sample is stained with antibodies or with antibodies or with probes, DNA probes labeled with fluorophores. After that, it is anchored to the gel with some kind of acrylate anchors. And then after that, it undergoes digestion. In this digestion process, some of the proteins are digested, and only the part that is linked to the gel remains. And then when you add water, the sample swells up to 4 times, and then you can image it really nicely. So I'll show you in a minute the images that I've obtained using this technique, but I want first to give you some tips and some notes that I have to this technique. So first of all, you should use small amount of samples, 8 to 10 samples, because you should remember this is a pretty tedious process to work with, to go through, and after that you want to image, and your samples are much larger than the samples that you started with. So you should take it in account, because it will take longer to image each sample. So it's better to start with small amount of samples. Second, you should use the double amount of antibody, of the first antibody and second antibody. And the incubation with the second antibody should be done for longer period, because for example, when I stain my samples, I usually use 1 up to 2 hour incubation with the secondary antibody, but here you should do up to 6 hours incubation, or sometimes I put it in the 4 degrees over weekend. And there are several fluorophores that you should use and several that you shouldn't use. For example, fluorophores don't work really good with this method and with any expansion method, because they bleach really fast after the expansion. So you should use these ones from this list. And for some reason, I don't know why, but Alexa 647 also doesn't work. So after you stain your sample, you do the anchoring. In this protocol, I anchor using MANHS, which is the ester that is bound to, from one side it has a acrylate group, and from the other side it has the ester group, and it binds to the from one side to the proteins, from the other side to the gel. Next step is the gelation step. So for the gelation step, I build these bridges using slides. So it's regular slides, 2 cover glasses that I glue using the super glue. In the middle, I put my sample embedded in gel and cover it with cover slip. So in this case, you just create this kind of a gelation chamber, because different protocols have different techniques to do this, but I found that this one is the easiest one to do, because in some protocols I saw people making some kind of wells, and it's more difficult to my opinion. So this was the easiest one, and it worked fine for me. And also, if your sample is thicker than 1 cover glass, you can put 2 cover glasses, one on top of each other, so your cavity will be larger for your sample. And this also allows you to image your samples before the expansion. The imaging step is really important because then you can compare your sample before and after expansion and evaluate the expansion rate and also the distortion rate of the sample, which is really nice. And after the gelation is done, you can store the sample, these slides in the humidity chamber using PBS. So this first part, the steps 1, 2, 3, can be done in like the staining, of course, takes time, but then the gelation step and the imaging can be done in one day. Next day, after you image your sample, you can start with digestion and expansion. So for this step also, it's really important to, if your sample is visible in this thing, it's really important to cut as close to the sample, because otherwise, if you don't cut it close to the sample, after the expansion, you won't be able to find your sample in the gel because it becomes transparent. So cut with the razor blade, syringe needle, or forceps the gel as close to the sample as possible, but leave some edges around it. And using a flat wet brush, you can transfer the sample easily from one place to another. So the next step of the digestion I do in these wells, but you can use any plastic plate or whatever is comfortable for you. And the digestion is done with the usage of proteinase K. And after the digestion, there is the expansion stage that is done overnight, and the next day you can already image your sample. For imaging the sample, I call this IBD wells. These wells have cover glass on the bottom, and these are the wells, like 8 wells, but they have different sizes. And it's really convenient because you can place all of your samples in the same slide. I cover the bottom part with polylysine, so my sample easily sticks to the bottom of this wells. And for the next stage, I just put it in this, transfer the expanded samples with brush to this IBD wells, and I can image easily with inverted microscope. So here you can see the example of the expanded tissue. So this is how the testis looks like. In my case, I used 2 proteins labeled fluorescently, MARF GFP and Fazioni tomato. It doesn't matter what are the proteins, but they are expressed in the mitochondria. So you can see the mitochondria, and you can see the nice structure that can be visualized after the expansion. So you can see in the first row the meiotic dividing cell, second row early stage of mitochondrial fusion, next later stage, the onion stage, and then the elongating sperm that only starts to elongate. So actually also it is possible to use DNA dyes in this stage. You can use just before the expansion stage, or also even after the first expansion round, you can use the host, for example, other DNA dye, and then you can also visualize your DNA. So this was a technique that is applied to the samples that are already stained. But there are several other techniques in which you first expand the sample and then you stain. Actually, I wanted also to show you some samples that I just found online. So this technique was already applied to bacteria and yeast. I know that many people have different tissues or different samples that they would like to work with. And I think the protocols are published for most type of tissues. You just need to look online and you'll find everything. So it looks like it works really nicely for bacteria and for yeast, and the stages are literally the same. The first you link the stain sample to the gel, then you do gelation, then you do digestion. The digestion is usually proteolytic digestion, and then you expand. So the same is for yeast. So this was staining before the expansion. But there are several techniques that apply staining after the expansion. So most of them are based on the magnified analysis of the proteome expansion technique. In this technique, you first fix the sample, you bind it to the gel, digest it using usually SDS and heat and some other chemicals, and then you expand it, and then you stain after the expansion. So the technique that I have experience with is the ultrastructural expansion microscopy, which allows you to expand the sample up to 8 times. This technique, you first fix the sample, anchor it using, if you remember in my previous slides, there I used the MANHS ester for anchoring. So in this case, you use formaldehyde and acrylamide anchoring, gelate, and then you denaturate the gel using heat and SDS, expand it 1 round, stain it, and then there is a second round of expansion. So this allows you to expand the samples up to 8 times. In my case, I wasn't able to expand it up to 8 times. It was around 5 times maybe, but still it was okay. But the problem is because of the chemical and heat digestion, denaturation of the gel, some of the antibodies won't work for this technique because the epitopes change their 3D structure, and some antibodies won't recognize the proteins. So for this technique, for this technique, it is easier to use. It is advised to use antibodies that probably would work for Western blot, because in case of Western, we also denaturate the proteins before. Also, this protocol was also published, and it has different anchoring and different fixation method for different organelles or samples that someone would like to visualize. For example, for mitochondria, the one that I wanted to visualize, I used PFA with glutaraldehyde. And if someone wants to visualize microtubules, it is done using methanol. And here you can see optimization for each one of the organelles or even small organisms like toxoplasma, I think they did. Yeah, toxoplasma. So here are the examples that they show in their paper. Here you can see the phage that was expanded, human cilium, and basal bodies, toxoplasma, and mitochondria. So it can also be applied to a variety of samples. And this protocol is also pretty easy. It takes about 2 to 3 days to finish. And it's not very complicated to apply. Next, so what's the advantages of post-expansion staining versus the pre-expansion staining? And when you stain your sample before the expansion, some of the epitopes are masked because just of the physical sites of the sizes of the antibodies that bind to these epitopes. And for example, this epitope won't be able to bind to the antibody. And after expanded, it's accessible to the antibody to bind. And also there is this thing called the linkage arrow. So for example, we have this microtubule that this is 25 25 nanometers in diameter, and it's bound with the first antibody and second antibody. So the calculated size of each one of this first and second antibody complex is 20 nanometers. So together is 64 nanometers. After expansion, 65 nanometers 4 times, you get the size of 260 nanometers. And this doesn't represent the correct proportions of the microtubule. In contrary, when you have this microtubule and it is expanded first 4 times, so it's 100 nanometers, and then you bind it with antibodies, the size is only 140 nanometers, and it's more accurate, and you get better resolution and better picture in this case. Another technique is, this expansion method, the ultrastructural expansion, is also discussed in this really nice workshop given by Andor. You can find it online, and it's also appears in the group's document. Gabrielle already put it there, so I really recommend you to listen to this workshop because it's really informative and it has a really good explanation. And it has really nice explanation about the different expansion techniques. That it compares between several techniques, and also has a nice samples demo that you can see. Actually, for my imaging, I also use under a microscope the spinning disc dragonfly, which is a really nice confocal spinning disc. Because the samples are so large, so the amount of data that you need to collect is enormous. And it's better to use a fast microscope than the slow one. So next technique is the pan-expansion microscopy technique, which allows you to enlarge the sample up to 16 times. So first, as in the previous protocols, we fix the cells and embed it into gel. And next, the gel is denaturated, and during this step, it's already expanded. And then there is the first expansion, which is done overnight, and we get the 4 to 4.5 times expansion. Next, the sample is embedded again in one gel and another gel, twice embedded in 2 gels, so it's 3 gels in total. And then there is a denaturation of both gels, the first and the second gels, and the sample is stained and expanded. So in this case, you can use pan-proteome staining, which is really nice, that it allows you to visualize all of the organelles or different parts of the cell without using any antibodies. So if you're looking for different architecture of the cell and you want to look at the different parts inside of it, it's really a nice option. So here, how it's done, the dye is called an HS ester. It binds to the primary amine groups on the proteins. And on the other side, it has usually the fluorophore. And then it just stains everything. So here in this image, you can see the staining before the expansion. So you don't see much, just see mostly the background. We actually applied it also to 4x expansion, the regular one, after the staining and everything is done, and just before the expansion. And here you can see the image of the 16x. So you can appreciate how nicely, for example, here the mitochondria is visualized using this this dye. So let's see sorry, this one jumped. So here you can see the images that I obtained using this technique, the ultrastructural expansion. So you can really appreciate how nicely I can see different parts of the cell and the architecture of them. You can see the nucleus with both of its membranes. This is the Nebenkern, the mitochondria that I look at. And it's really, really nice, but the downside of this technique is that most of the antibodies don't work after this extended denaturation steps. In my hands, I was only able to stain with certain anti-GFP antibody to visualize membranes of the mitochondria, because I have one of my proteins is fluorescently tagged with GFP. But I wasn't able to apply other antibodies, but in paper, in the original paper that described this technique, they have several other antibodies that they claim that work fine for this technique. So I also consulted the authors of the paper, and they gave me some really nice suggestions of what I can do improve, what I can improve in my in this method, in using my samples, and it worked really nicely, but without staining. So here you can see the comparison of two expansion techniques that I used to the EM images. This is the 4x expansion. This is the pan expansion. So it was claimed to be up to 16 times. In my hands, sometimes it works better, sometimes it works worse. So in this case, it was 12 times, but in some cases, it was up to 15 times X expansion. So I guess it depends on the materials that you use. You should use really fresh materials and don't wait too long in different stages, because this is also the reason why you shouldn't use too many samples. If you're too ambitious, you use too many samples. So sometimes you mess it up and you get with the less desired result. So here you can see the comparison between the EM image and the expanded images. And you can appreciate that the architecture is pretty similar, and there is not much distortion of the samples. So it's really nice to see that we get similar result with the expansion technique. So just to summarize, I told you about pre-expansion staining versus the post-expansion staining techniques. So the samples are pretty much everything that you can think of. Sometimes it can be applied even to some proteins some proteins or protein complexes in the solution, and cell culture, whole tissue, everything that you can think of. So for example, this technique, the pan expansion, the microscopy technique, was originally applied to tissue culture, but after that they developed a protocol which allows to visualize brain slices. And the antibodies, the pre-expansion staining, you use the same antibodies as for regular immunohistochemistry. As for post-expansion staining, not many antibodies work, so you should use the ones that work for Western blot and hope that they will work. In my hands, when I use the ultrastructural expansion, I used this red antibody, which worked really fine. The digestion in pre-expansion staining is enzymatic, and post-expansion staining is chemical digestion and heat denaturation. Final expansion factor for pre-expansion is 4 times, for post-expansion up to 16 times. The time, the pre-expansion staining is less time consuming, it's only 2 to 3 days, while post-expansion staining takes longer, especially the ultrastructural expansion, especially the pan expansion staining, pan expansion protocol, which takes longer. And the final imaging as much as you expand the sample, it becomes thicker and larger, so it's more difficult to visualize it. So in 4x, it wasn't a problem, but when I did the 16x, I have to slice the sample in the middle to make thinner sections to visualize it. So it's also something to take into account, and also the sample becomes really, really large, so it will take longer to visualize it. So that's it. This is my group. This is our lab. This is my boss, Elia Rama, and we'll happy to take questions.
Thank you, Lena. That was great. I'll let people unmute themselves if they want to ask a question, or you're happy to write them in the chat otherwise. What people are thinking about that. I have a few questions for you. One of them was like, do you need to have any sort of like fiducials or beads in the sample to know exactly how much it expands, or it's just by known structures that you were calculating?
Yeah, I calculated because I know that my mitochondria, this large mitochondria, is 6.7 microns in diameter. I can calculate it. After that, compare the size. I also visualize it before and after expansion, because my samples are fluorescently labeled even before expansion, so I can go to the microscope, visualize it, and just find the same region in my sample and visualize it and see. So it's really easy, but also some people just measure the size of the gel before and after. But when you use pan expansion technique, you slice the gel several times because you have several gelation steps. So you slice the excess gel, so it's more difficult. So beads, I don't think the beads would work because well, they probably won't expand as well as the sample. I don't know what are they made, but I guess they won't expand to the same extent. So usually it's better to have something in the cell that you know the size of it. For example, the nucleus or the centriole or whatever, and then you can measure it before and after.
There's quite a few congratulations in the chat. And also a question from Matthew Rowe about, was wondering if the expansion is uniform across all cell structures, or do you find that certain structure expand differently?
Okay, so the lipid droplets don't expand. And they don't survive really good the protocol because of the denaturation steps and all the and others. In my case, for example, I have this 2 millimeter long sperm, right? And it has the axoneme, which is a microtubule actually. And a part of it breaks in like small chunks because it's very rigid. So when it's expanding, in some cases, it doesn't go well through digestion and it breaks in parts. But yeah, some things, that's why they also use different fixing techniques for like different fixing reagents for different structures that of interest. But usually it's pretty uniform, but some structures, the rigid ones, the ones that are really dense, sometimes they can break down into parts.
Thank you. The other question would be like, if you're like a newbie and you just want to try one of these techniques, which one you would go?
Okay. Yeah, I forgot to mention this. You should definitely start with the simplest expansion technique when you stand your sample before the first one that I discussed, because it's pretty straightforward, and also you can visualize the sample before and after to evaluate the expansion rate and to see if it it's not distorted, and to compare it. So it's really, I would start with this one. I would start with the first one, and also really when people ask me to teach them expansion, some from other labs, I advise them to start with the first pre-stained sample expansion.
Okay, good. There's no more questions in the chat. The only other question was if you would be willing to share the slides with some of the attendees. That's another question I'll read after. But I might ask you that by email later. I wonder what the minimum size of the sample you can use worth this technique. So what's with, sorry, that was an autocorrect. What's the minimum size of the sample? Yeah.
I think it was applied to protein complexes, so I guess several nanometers is okay also. Single cells are fine because it's really developed for any kind of sample.
Yeah, I guess the question becomes then a little bit the handling of the sample.
Yeah, okay. So that's why you have the gel, so you can cut it to the size that you are interested in. Let's say testis samples. I would, well, you can't cut it smaller than something that you can see with your eyes, right? So yeah, as small as possible, but you won't cut it smaller than something that you can lift with the brush.
There's another question. Do you experience photo bleaching problems during imaging in water, and how long can you store the samples in water?
Okay, the samples can be stored up to 1 week in water. And also you can expand them gradually. You can store some of them in PBS and apply to the ones that you want to image now. You can expand them and start with them. In my hands, it was good for up to 1 week. Yeah, it depends on the fluorophores. As I said before, the Psi5, for example, for all the other Psi fluorophores bleach fast. And I don't know why, but for some reason I ordered NHS the one that for proteome staining. I bought one that is conjugated to Psi5, so it bleaches much faster than other fluorophores. So yeah, you have, of course you have photo bleaching, and to start with, the signal is much weaker than you would have in regular samples before the expansion, because the distance between the fluorophores is larger. So you would have to use stronger lasers and longer exposure times. So yeah, it will bleach eventually, but I don't think it will bleach more than the regular fluorescent stain sample.
But there's no anti-fading or anything like it in the solution, in the solution.
Sorry?
Anti-fade reagent, no? On the solution, no. It's just PBS.
No, it's just PBS. No. You can store it in PBS and then in water. After it's expanded, you have to store it in the water because you don't want it to shrink back.
There's another question about, can you expand paraffin embedded histology sections?
No, because it's in paraffin already. So
if you deparaffinize,
so what is usually done? You instead of embedding it in paraffin, you embed it in gel, and then you can slice it using a vibrotome, and then you get thin sections. For example, for the brain tissue, they did it this way. They first fixed it, then they sliced it, and then they fixed it again and embedded it into gel. And after expansion, it can be sliced again. So instead of paraffin, you just use the gel. That's it.
I don't see any further questions in the chat. Yeah, we wanted to thank you. There's a lot of congratulations in the chat about the great talk, and it's great to get some practical tips because
Yeah.
it's easier to read the papers and it's like, oh, but so from someone that has done it, that's great help.
Yeah, just really just jump into the water and try it. It's really not, it's not very complicated. You just need to try to see how it works.
Thank you, and thank you everyone for joining. Remember to put your attendance in the Google Doc we have, and if you have any questions, you can post it in that Google document. Make sure you put a contact, email, so people from Canada, people from here can help each other. And if you want to present, or you know someone that is happy to present, we're probably going to have our next meeting in another 2 months. So you can just let me know. I think with that, we are going to finish the meeting. And thank you again, Alina. That was great.
You're welcome.
Thank you for waking up so early.
So, yeah, and anyone who have questions, feel free to contact me. I'll be happy to help.
Great. Okay. Thank you.